Well, why don't I just get started with the introduction, and I don't want to take any time away from Dr. Abreu. So hello, everyone. I'm Johnny Hermazdi. I'm the Chief Medicine Resident here at the University of Washington, and I'd like to welcome you to the first Department of Medicine Grand Rounds of the new year. On behalf of everyone in the department, I want to express my gratitude to everyone for the hard work and sacrifices you've made over the past year. As we were talking about, 2020 was a year of unprecedented challenge and disruption, especially for the Department of Medicine. And I'm continually amazed by the resilience and grace on display at every level in this institution in weathering this storm. So while we look ahead to this new year with optimism and hope, I do want to take a moment to say that the events of this week have reminded us that an incredible amount of work remains and more challenges still lie ahead. And in light of that, I want to reaffirm our commitment to progress in science and medicine, in equity and justice and peace and respect for all. So on that note, I couldn't be more excited about today's speaker, a titan in the field of gastroenterology. Dr. Maria Abreu from the University of Miami Miller School of Medicine is a physician scientist who specializes in inflammatory bowel disease. She completed her medical degree at the University of Miami Miller School of Medicine, and her postdoctoral training included an internship and residency in medicine at the Brigham and Women's Hospital in Boston, a clinical and research fellowship in gastroenterology at UCLA. She was an associate professor of medicine with Mount Sinai School of Medicine in New York City and the head of the IBD Center there before returning to her alma mater in Miami. She currently directs the Crohn's and Colitis Center and is a professor of medicine and also a professor of microbiology and immunology at the University of Miami Miller School of Medicine. She was honored in April of 2018 with the Provost Award for Scholarly Activity and appointed in 2018 as well to serve as Vice Chair of Research for the Department of Medicine. She is internationally renowned with over 20 years of leadership in basic, translational, and clinical research, and has been a council chair of the American Gastroenterological Association Institute Council. And her most recent appointment is as the deputy chair of the International Organization for the Study of Inflammatory Bowel Disease. She was recognized as the new counselor at large member of the AGA governing board for 3 years in 2019. And she recently received both the Helios Lifetime Disruptor Award and the Sherman Prize by the Bruce and Cynthia Sherman Charitable Foundation, celebrating and recognizing her outstanding achievements and innovations in IBD research, including over 100 peer-reviewed publications and editorial board contributions to too many journals to name. So with that, I am so excited. We are all excited to have her here with us today. Dr. Abreu, thank you so much for making the time to share your knowledge with us. And on
you made my day. You made my day.
Oh, good. On a brief logistical aside, we've tossed out the Zoom Q&A feature for today's Grand Rounds. Please make liberal use of that chat box. I'll be watching and moderating for you, Maria. And please go ahead and get started.
Awesome. Well, what a pleasure to join you, Johnny. What a lovely introduction. You didn't have to do that. These people have nothing else to do at lunchtime, I suppose. So they're stuck with me. They're stuck with me for the next hour. Maybe some of you who logged in early saw that I've known your boss, Barbara Young, for a long time. And I'm so pleased for you that you have such a terrific leader that has joined such a great institution. And as I said to Johnny, some of the privilege of some of the advances, I suppose, that have occurred as a result of this pandemic is my being transported, from this, the bottom right hand of the country, as far away from Seattle as you could be, all the way to you, and saving not only time, but I think Barbara and her department a lot of money. So I think all of that is good. I hope I didn't put too many slides, Johnny, and I'm gonna, look down at my watch occasionally, because what I really am hopeful of is to have more time for questions, because, I could talk all day, and I think that's really not the purpose. These are my disclosures. I'm aware that I'm talking to a general medicine audience, that apparently not absolutely everyone wants to go into GI. And as I was saying to Johnny, in fact, I think many people nowadays may even want to go into pulmonary critical care and ID, which, again, all of these are some of the other side effects of the pandemic. But what you can see here is it kind of what you'll remember from medical school, which is that, ulcerative colitis. Oh, I wonder what that is. Let me I don't know if you're seeing that on my screen. Sorry about that. Let me just go back into this mode. There's a weird thing started happening, kind of like I was watching um, um, like a closed captioning of what I was saying. Anyway, so ulcerative colitis, as the name implies, is inflammation that's limited to the colon. It has another principle, which is it's limited only to the lining of the colon, so not even the full thickness of the colon. And the other rule of engagement for ulcerative colitis is that it always starts in the rectum, and in a continuous fashion can involve as much of the colon as it wants to. So there are some people that only have it in the rectum, but at the other extreme, there are people that have it in their entire colon, which we refer to as pancolitis. And then there's this other disease, Crohn's disease. That's another inflammatory bowel disease. The most common scenario is that people have a combination of small bowel or ileal involvement with some amount of their colon involved. But there are people that just have it in their small bowel. There are people who just have it in their colon. And there are people that, really fit a little bit of both and are termed indeterminate colitis. These are some, endoscopic pictures just to give you a sense of what Crohn's disease looks like. You'll remember from medical school that Crohn's disease is patchy, and that's very well exemplified here on this extreme left-hand picture, where you see normal appearing colon and then someone took a bite out of the tissue, or what we might have seen as cobblestoning if this was a small bowel series. Not that anyone in this audience has ever seen a small bowel series, because you're all too young to have ever seen a small bowel series. You don't get quite that same effect when you do MRI enterographies or CT enterographies. And then over a longer period of time, one of the complications of Crohn's disease is that inflammation in certain people begets fibrosis, and these waves of inflammation and fibrosis can eventually lead to a significant enough stenosis that patients require surgery to remove segments of intestine that have become obstructed. This is ulcerative colitis. My analogy for ulcerative colitis is that it has the appearance of road rash in the colon, as if you took sandpaper and rubbed the inner lining off of the intestine, and you can sort of imagine what the symptoms might be like in someone like this, or in Crohn's disease, which is that patients have malabsorption and maldigestion that results in diarrhea. They often have blood in their stools. That's particularly true in ulcerative colitis. And as Barbara is interested in cancer, and I am as well, in colitis-associated cancer in those patients that have had chronic inflammation of their colon. What causes inflammatory bowel disease? A lot of work has been done to identify the genetic susceptibility that results in IBD, and there have been over 200 loci that have been described, most of which are in common between Crohn's and ulcerative colitis, which just that observation alone I find fascinating, given that I just showed you pictures that look sort of very different from each other in their disease expression, and yet at a genetic level, the infrastructure of these two diseases is quite similar. That that some medications, can exacerbate the disease. For this talk, I'm not going to tell you all the different treatments there are for the disease. I'm not aware of any condition where patients don't think that stress caused it, and I do think that very, very severe stress could be at least a contributor to flares of inflammatory bowel disease. Diet, we'll get back to what the role of diet may be as a treatment opportunity that is only relatively recently being explored. Smoking, I think you'd know this, the GI people in the audience would know the dichotomous role of smoking, that it very much worsens Crohn's disease and is protective from the development of ulcerative colitis. And then, of course, the microbiome in every field of medicine now, people are interested in the microbiome. As far as I'm concerned, they're Johnny-come-latelys, because the whole field of microbiome, or a big importance of the microbiome from an early point, came about from the study of patients with inflammatory bowel diseases. So what have been some advances in our field? Well, IBD, I would argue, has led the discovery of genes in complex genetic disorders. When we got done with single gene diseases, I don't say get done with, but when it became trivial really to find these single gene diseases, then the real challenge became, what about these complex genetic disorders like inflammatory bowel disease? And so there's a lot that's been learned, most recently with whole genome sequencing, and in every phase, whether it's GWAS studies, whole exome, and now whole genome has become the standard. And although I don't have a slide on it, I think there are up to, at the Broad, about 70,000 whole genomes that have been sequenced in IBD patients. The whole microbiome initiative, whether it's the one being led in the US or the one that's being done in Europe, that is for healthy people, has also come about really as a result of studies early on done in the microbiome of IBD patients and comparing IBD to people who don't have IBD. Trying to discover what causes IBD has led to very high quality early cohort studies. I think that I have sort of one or two slides to deal with these topics. And then I think that diet intervention studies are going to be the new very big opportunity to complement some of the therapies that are available for IBD currently. That would be one scenario. And also, I think probably to help prevent or mitigate disease expression in high risk groups. So why do we want to know what causes the inflammatory bowel disease? Well, I gave a provocative title to the talk about whether or not we're getting closer to curing these diseases. And how do we cure a disease if we don't know what causes it, right? Is it possible to cure a disease? Can studying the genes, the microbiome, the environment identify the cause or lead to better therapies? I think you probably have all drunk the Kool-Aid and think that the answer is yes to all of those. Are ulcerative colitis and Crohn's disease at all related to each other? The pictures I showed you, other than being colonoscopy pictures, look very different. And again, at their extremes, they can be very different, but kind of in the middle, they could look very similar to one another. And is everyone's disease unique, or are there enough commonalities to find a universal point of attack? So to flip it around, for example, things like the PD-L1s or the checkpoint inhibitor type therapies for cancer are, in a sense, agnostic to the cancer, right? They're boosting an immune response broadly to neoantigens that might be present in someone's unique tumor, so that even though you don't know all the characteristics of that person's unique tumor, maybe there's a way that you could, universally treat the process. Okay. What chronic diseases are curable? Actually, for all intents and purposes, for most of the things that you and I are doing, including COVID-19, we aren't curing COVID-19. We're trying to eradicate it by having all gotten vaccinated. In our world, for example, hepatitis C, it wasn't until the etiologic agent of it was found that there were cures for hepatitis C, and then that becomes easy if it was one pathogen, right? If we knew that IBD was one pathogen, we could try to eradicate that one pathogen, however way we do it. HIV. It's a semantic issue if we view HIV being cured. Certainly patients that we care for have no viral load on direct acting retrovirals, but if they stopped it, obviously the HIV would begin to replicate again. People talk about curing cancer. I think that that's a kind of a mistaken expression, in that cancer isn't one thing, right? And so, yes, of course, there are people who are cured, thankfully, of cancer, whether it's through surgery or through chemotherapy. And I think that, and I think the oncology world is in some ways leading the field of taking on personalized approaches by sequencing tumors. But on the other hand, we're all still limited. We are in IBD as well, with a defined list of things that we can use, that we have in our toolbox, right, to treat these patients. So that, yes, of course, it's helpful to know what the underpinnings are of someone's cancer, but it's only useful if you actually have something to target that pathway. So one way to cure the disease that is inflammatory bowel disease is to prevent it altogether. That's cheating a little bit, but nevertheless, it's no less laudable to prevent the disease from happening altogether. And in that rubric, we have different studies that are trying to figure out what is the earliest point that we could identify that someone is destined to develop IBD, so that we could intervene in some way to prevent them from developing IBD. Or could we let someone express the disease and have it be a one and done, that they have one flare they present with, and then you then intervene with X and prevent that person from having any more flares of disease. The GEM project, which is genes, environment, microbiome, is a project that started in Canada, and it's a project in which I told you these diseases have genetic underpinnings. People with Crohn's disease who first degree relatives of people with Crohn's disease, of course, are at a higher risk of developing Crohn's disease. And the conception of this study, which is a labor of love, is to collect blood, urine, stool, of course, on people who are first degree relatives of Crohn's patients, and then follow them over time for those patients, for some of those people to ultimately develop Crohn's disease. This probably would need to be updated. Look at this, they collected 5,000 people to have 70 people develop disease. So if you're a first degree relative of a Crohn's patient, you could sleep easy, that the chances that you'll develop Crohn's disease is low. It's higher than people who don't have it, but it's still quite low. But at least in these people, as that number goes up, we will have known what their microbiome, what their intestinal permeability was, what their immune parameters were in advance of them developing the disease, and that could again highlight people that we could intervene on if we had a good intervention. This is one of my favorite studies that was published in Gut in 2019. Don't read so many words. Let me just tell you the story. When does IBD sort of start, or when do people become at risk? So this is an amazing study that my friends at Mount Sinai did. They got the buy-in of an Orthodox Jewish community to collect stool from moms who had IBD or didn't have IBD, and the meconium from the newborns born of women with Crohn's disease or not. Okay. And to cut to the chase, the microbiome of IBD patients, of course, is different. That's not like you didn't have to log in for that. But amazingly, the meconium, which we would think is like I think I grew up thinking it was sterile, right? The meconium of these infants born of mothers that had Crohn's disease was clearly different than the meconium of babies born to mothers that didn't have Crohn's disease. I find that, like, in principle, amazing, shocking, and I don't think it's on this slide, but these babies born of mothers with Crohn's disease even had elevated fecal calprotectin. Not elevated, but higher than the babies who were born from healthy moms. Crazy stuff. So I'll get back to this conceptually, because again, I should tell you that the reasoning for doing this study is that if it's your mom who has Crohn's disease, the chances that the baby will have Crohn's disease is higher than if it's the dad. Some of you are probably thinking cynically, that's because you never know who the dad really is. Not kidding. All right. The other kind of cool study was done led by, again, colleagues at Sinai in New York, where they looked at serum that had been collected on military personnel. These are young people. This is a disease that occurs in young people. They probably didn't have symptoms when they joined the military, or at least they weren't screened out of joining the military as a result of it. And they collect serum on these military personnel. I don't think it's only Army, every 2 years. And so that serum was banked. And then some of those people developed ulcerative colitis or Crohn's disease. And they applied a variety of different either tests that are known, serologic markers. Maybe some of you in the audience know that patients with Crohn's have antibodies against Saccharomyces cerevisiae and other microbial, gut microbial antigens. Those start rising in advance of the person developing Crohn's disease, and the higher those values of antimicrobial antibodies, the closer in time to the more likely is that they're going to be diagnosed with the disease, and the more severe the disease is. Curiously, ulcerative colitis doesn't show any of these markers, right? And that's also fits my bias that Crohn's disease could be kind of like flipping on a light switch, that they're fine until they're not fine. And similarly, in someone with ulcerative colitis who's in remission, something that throws them off. It could throw them off, and you can't even get them back with medical therapy. The pathogenesis of IBD is multifactorial. I showed that to you conceptually in terms of, diet and physical stuff and the environment, but I just wanted to show you kind of at a gut level, the various tiers, if you will, that of pathogenesis, but importantly, I view as potential points of targeting therapy. And so the microbiota, I have other slides to show you this, that what is healthy, just like for a healthy country, is diversity and more density of bacteria. Curiously, IBD patients have less diversity of bacteria. A little bit is a chicken and egg issue, because if you cause inflammation, you can also decrease diversity, and some of these are some of the things that might contribute to that, right? More hygiene, use of antibiotics, which is also related. More antibiotic use in young kids, more IBD later. Then there's the level of the mucosal immune response, that this activated mucosal immune response, where there might be modules of cells that have become rogue and together are cooperatively probably reproducing a program to stop a pathogen, but of course having deleterious effects. In addition, actually, genetically, there are signs that the epithelial barrier is also defective in people with IBD. We'll include in that Paneth cells, which are these little antibiotic machines at the base of small intestinal crypts, but there are Paneth-like cells also in the colon. They not only make naturally occurring antibiotics, but they also make factors that are important for maintenance of the stem cell niche. So there's a lot of important stuff, I think, going on with the epithelium that, at least at present, is not specifically being targeted in any of our therapies. And then these inflammatory cells, whether they be T cells or monocytes, travel to the intestine, or are able to access the lamina propria of the intestine through homing receptors that are on the surface of the lymphocyte, for example, that need to dock on the endothelium of the intestine in order to access the intestine and cause inflammation in this case. And there are various therapies that are directed at disrupting that homing of these inflammatory cells. I will also use the word dysbiosis in this conversation that I'm having with you. That's a kind of a loosey-goosey term, because even what is defined as normal is not is imperfect. So, but I like this kind of I like this drawing of the gut, and you can see the villi. I mean, the small bowel is absolutely gorgeous and so logical. And overlying that single layer, the plan B to keep pathogens out is, of course, to have a nice thick layer of mucus, one sort of impenetrable layer, one that we're allowing bacteria to kind of interact. We eat food. Maybe you're eating food now, and thankfully I'm not watching you eating your food, because that would make me hungry. You eat fiber, for example, that is insoluble or that's not metabolized by any of our mammalian carbohydrate enzymes. This fiber then is substrate for bacteria to generate, among other things, short chain fatty acids. Butyrate, of course, is thought to be beneficial. But in some cases, I think that many of us and many patients with IBD are missing some of the actual bacteria that are able to metabolize fiber into butyrate. So they're completely missing organisms in our gut, which is problematic. Okay, on this slide, maybe all I want to say to you is that yeah, okay, dysbiosis, this thing that is just different than health, has been found in IBD. It's also been found in a lot of, like, pick a disease that you might be interested in, and you will also find that people have dysbiosis. There are even people studying depression and things like that, and there are metabolites being generated by the gut that can have central effects. We don't have to go that far. Some of you take care of patients with hepatic encephalopathy. That's a perfect example of how gut metabolites are actually having neurotransmitter-like effects, right? So in the world of IBD, a couple of themes that have emerged include the fact that there's a subset of patients with Crohn's disease that have adherent invasive E. coli. that has a specific definition, as a pathobion. I might, if I have time, explain that a little later, and maybe targeting that would be beneficial. And then there's a decrease in obligate anaerobes, and one particular obligate anaerobe, this F. prausnitzii, seems to have beneficial effects, like in other words, anti-inflammatory effects. And investigators are trying to figure out, can we colonize people with this, and would that have a salutary effect? Okay, this is a super old school slide, a very old school slide, and maybe only Dr. Young and I remember this slide. In the early days of genetic models of making recombinant genetically engineered mice, overnight, when you start playing around with various genes in like central genes in immune response, like interleukin 2 and interleukin 10, or T cell receptor alpha, the alpha chain of the T cell receptor, these animals spontaneously developed colitis. Now, I'm thinking that wasn't the intent of the people, the investigators doing this kind of work, but yet overnight we had a burgeoning of these animal models of IBD. So that was very interesting. Lots and lots of work have been done in those models. The next interesting chapter in that story was that if you then were able to rederive those mice in a germ-free state, they no longer had inflammation. Now, to us, this seems like duh now, but the T cell mafia used to think that everything was only related to the host immune response, and wouldn't consider that the microbiota could be playing a role in at least activating that inflammation and possibly perpetuating that inflammation. And so that was a long-winded way of showing you, kind of this slide, that in health, I told you diversity of the microbiota. We all carry some genetic risk. We must all have some of the polymorphisms that result in IBD, yet most of us don't have it, just like even most of the first-degree relatives of Crohn's patients don't have it. Oh, sorry about that. But then what comes first? Does inflammation start increasing? Like something happens that causes these cells to start, again, becoming rogue? And does that then cause dysbiosis? Or do you have some subtle degree of dysbiosis because you took a course of antibiotics or whatever, or you went swimming in the ocean, and that leads to an increase in inflammation? And then once there's inflammation that's increased, almost coincident with there being inflammation, there's a decrease in microbial diversity, more mycobiota, in other words, fungus, and more virome diversity. And then curiously, perhaps not surprisingly, any effective treatment of IBD that reduces inflammation will start bringing back to normality the dysbiosis seen in IBD patients. Not all the way, it's hard to get it all the way, but it will make a substantial effect on the dysbiosis. We, like many other centers that take care of IBD patients, have a biorepository of samples where we have very detailed annotated data now on 3,000 patients with IBD, where we've collected these patients, DNA, RNA, and blood, serum, and on more than maybe 60% of them, we have biopsies that we've taken in a protocolized way from the ileum, from the ascending colon, from the sigmoid colon, as well as stool and serum at the time of colonoscopy. So it's a sort of a treasure trove with a lot of very carefully annotated data in REDCap. And we did a prospective study to try to understand, if you've ever looked at some of these microbiome papers, they're seizure provoking because it's a lot, it's now, well, it's a lot of deep sequencing data to identify genetically what microbiota are present. So it becomes an exercise in mathematics to identify all the different organisms that are present and to kind of compare them to normal people. The issue, of course, I shouldn't say the issue, but an issue when you do any microbiota work that's been exemplified. Actually, there's a recent paper, I think, in Science that deals with this problem of the noise between individuals and the noise between studies, because most of the studies that are done on the microbiome are on the small side, especially in disease states. And so we wanted to test the hypothesis that, sure, people had been studying up until this point stool microbiota, maybe mucosa-associated microbiota, but we reasoned that if we were going to, if patients with IBD had pathobionts, or what I call situational pathogens, in other words, organisms that maybe to you and me wouldn't be pathogenic, but perhaps in the right sort of genetic context would be, that those bacteria, and we only looked at bacteria for this particular study, would be more likely to have crossed the barrier, the epithelial barrier, and be phagocytosed by mononuclear cells, macrophages, dendritic cells in the gut. And so what we decided to do was to look at the microbiome in biopsy samples, where we either looked at the entirety of the mucosa-associated microbiome, like, if you took a bite of tissue, it's kind of everything that was on the surface and everything that came in that bite of tissue. That includes the lamina propria. And compare that to the microbiota in isolated professional phagocytic cells of the lamina propria, right? So the professional eaters of bacteria that come across the epithelial barrier, we reasoned would be found in phagocytic cells. And one marker of phagocytic cells is CD11B cells. I won't show you the data, but these CD11B cells are a combination of macrophages, dendritic cells, and in active inflammation, also neutrophils that are present. And so we isolated these CD11B cells, we sequenced them. And what's nice about this particular study is that each person could be their own control, right? Because you have, what we're comparing here is that what's in the mucosa compared to what's in the lamina propria of the same individual. Hopefully that makes some sense. In the blue and the green are the microbiota. This is the community structure. If you're used to looking at these principal component analysis, like a very broad view of the community of bacteria that are present. In this case, it's in the lamina propria, right? These CD11B cells, versus in the mucosa, which are in the pink and gray. And just looking at it with, without knowing anything, you can see that the blue and green dots kind of are hanging around together, whereas the fuchsia and gray dots are clustering together, suggesting that what is living, or what can be sequenced out of the lamina propria, is a different population, or a skewed population of microbiota compared to what is living on the surface of the gut. And you can see that represented in a slightly different way here, that you can see proteobacteria is already known to be overrepresented in IBD. But yet that overrepresentation is even more accentuated if you look at lamina propria CD11B cells. Likewise, Bacteroidetes are underrepresented in IBD, and you can see that they're particularly underrepresented in the phagocytic cells in the lamina propria of these patients. And so what I think this could mean is that we're looking at a more distilled version of the dysbiosis in these patients by getting into like where the action is really happening. And the reviewers believed us. So you can see here, like kind of how we, how we have conceptualized it, which is that there's dysbiosis reflected in a deeper level of the mucosa than what we have been focusing on up until now. And so, and we hope to kind of continue this work. I have a talented graduate student in my lab who's trying to develop techniques to do dual sequencing, so we can identify simultaneously the microbiota living in individual cells that are in the lamina propria, by doing deep sequencing that allow us to deconvolute what bacteria were they, as well as what genes are being expressed by that particular cell at that particular time. Okay, so this is just kind of like a provocative idea. I've mentioned on a few occasions how genetically Crohn's and UC have a lot of overlap, yet endoscopically and and at the bedside they can look very different. And it makes me wonder if ulcerative colitis is a more generalized problem, like the epithelium has gone to seed, for example, possibly because it's deficient in certain metabolites that should have been produced by bacteria that are either missing or not getting the right substrates. Whereas a certain type of Crohn's disease almost looks like, what could possibly cause there to be an ulcer here, but leave this alone? Like, what causes this sort of patchiness? Is there like a tissue invasion? I mean, or some pathogen that's set up shop and won't move out of Washington. So not all I'll remind you that not all infections are curable. And so what if, because a naysayer could say, well, if we found a bug, we could just give someone antibiotics, and there have been studies directed at, say, mycobacteria to see if that would cure or treat Crohn's disease. The answer is it doesn't. But I could spend all day talking to you about why those studies, the great studies, still haven't been done. And part of the issue is that it's actually very hard to kill mycobacteria, as an example. So if you wanted to believe that kind of faction that thinks mycobacteria is the answer, how do you treat it? Likewise, this adherent invasive E. coli story, which is a really kind of cool story, where wouldn't it be great if we identified who had this AIEC and then treat those people differently? Now there's a study going on actually to try to do that. This adherent invasive E. coli, it isn't trivial to identify it because it's defined by this E. coli actually growing in macrophages. So it's not like, oh, like let's order that test and we'll get the results by the end of rounds. But I mean, it's not impossible, not impossible. So this is just this adherent invasive E. coli story where the bug is able to cross the epithelium and is taken up by activated macrophages and kind of festers and causes disease. Now there's a trial to see if you find people who have it, the 40% that have it, treating them with Cipro and rifaximin to see. So we'll see. But the thing that I find really kind of interesting is this novel approach that maybe we can use phage, like bacteriophages that are specific to like organisms. So for example, let's say our research found another organism that we think, 20% of Crohn's patients have this organism. We think that this is the one that's causing the problem. And we don't want to just give bludgeon type antibiotics, right? Because antibiotics are bad in other ways. Maybe we can make, through CRISPR or other things, a phage that will just delete that particular type of bacteria. And that's not beyond the pale to imagine that. In this study, I think this actual study was in 2018. I was actually trying to find the reference before our conversation here. But this is looking at mice. These mice were infected with different strains of this AIEC, and they gave them a cocktail of a bacteriophage cocktail versus PBS. You can see that the mice that got the phage cocktail had a big drop in these in the CFUs of this AIEC. So that's a kind of a cute approach to saying, okay, we're gonna just target that those pathogens very specifically. So I like that. Let's see, Johnny. I think I'm okay. I don't know if everyone's asleep, but I'm okay. So can the study of immigrants edify the causes of IBD? And it's a curious thing. You may know, like probably other diseases that you might be interested in, that the rate of inflammatory bowel disease is really on the rise in newly industrialized countries, especially in Asia and Latin America. And so you can see here, I mean, whereas in the United States, in the population that is, born multiple generations here, it's kind of plateaued. It's now, I would argue, the immigrants to this country that are responsible for a rise in the rates of these diseases. You can see here, Crohn's, 11% increase year over year of cases of Crohn's and ulcerative colitis in Brazil and Taiwan and South Korea. All right, so that makes that point. We're here in Miami-Dade County. Miami is the capital of Latin America, and it's reasonably egalitarian in terms of the diversity of people from Latin American countries. The pack is still led by my tribe of Cuban immigrants or children of Cuban immigrants, followed by Colombians, and this, we've updated this recently, and this hasn't changed very much. This USA bar is to represent the fact that these are people that are, had to have both parents were from a Latin American country, not necessarily the same Latin American country, but born in the United States. We're seeing, we've seen more recently a blip of people that are coming from Venezuela. It's wherever there's a diaspora of people that we have a blip in Miami. this is a study done now several years ago by a young woman who at the time was a medical student. She's now a K awardee, Barbara, and kind of a rising star in IBD, Oriana Damas. And she described that if you stratified, if you do like a lot of studies have done in the published literature and just compare Hispanics to non-Hispanic whites, you'd miss the fact that for US born Hispanics, they're developing Crohn's disease and ulcerative colitis, kind of like, non-Hispanic whites and at a similar age. But if you look at foreign born Hispanics, they tended to have more ulcerative colitis and to present at a much later stage, like, they need to be in the United States for a bit longer. That's not explained by genetics. So if you look at the polymorphisms that have already been described for IBD and give those a score, a point score, a couple of things emerge. First of all, it is already known that Crohn's is associated with a higher genetic load for developing Crohn's disease. It sort of goes along with the fact that identical twin studies, if you have Crohn's, the identical twin there's a 50-50 chance of the identical twin having Crohn's, whereas it's much lower, less than 20% in ulcerative colitis. So there are probably more environmental triggers when it comes to ulcerative colitis, right? And so if you tally up all these different polymorphisms that people have that serve as susceptibility to IBD, Hispanics and non-Hispanic whites have a similar load. So that the observation that the rate of IBD is much lower in Latin American countries, in other words, has nothing to do with at least genetics at this level. We can talk all day about lots of genes that may not be discovered yet because these patients or these people have not been up until now included in most of the European-based studies of IBD genetics. So we wanted to understand, okay, fine, so it's not genetic. What about the environment? And so Cuba lends itself to that experiment in that you have an island. It's been under, communist rule. People can't easily leave, and people aren't coming in to enrich the island with genes from other places, right? Not a lot of people wanted to move to Cuba and set up shop there. And their diet, at least in Cuba, is relatively monotonous. Again, we can talk about that another time. And then they come to the United States in different periods of time. So how long is it taking them to develop IBD, and what kind of IBD are they developing? And so I don't even know if many of you know, this is the back of Fidel Castro, when he took power in 1959, talking to hordes of people in the Revolutionary Plaza in Havana. In 1980, there was something called the Mariel Boat Lift. Many of you were probably just blasticists or not even in 1980. And so there was a big wave of people leaving Cuba in 1980 during the Mariel Boat Lift time. And then in the 90s, people were going hungry. The Soviet Union stopped supporting Cuba as it had for decades because there was a false economy. And people would make a raft out of anything. This looks like a proper boat kind of thing, but they would come over in anything that they could, that would float. And that stopped in 2016. Obama said that he would no longer let people just become automatically citizens in a year. The Pope visited Cuba, Obama visited Cuba, and then of course, we've again gone back to having this very radically I don't want to say anything prejudicial, but a very extreme policy against Cuba, which is only harmful to the people in Cuba. But so it set up it sets up this experiment. Now that valve is closed, there are no more immigrants. We're not a lot of immigrants coming from Cuba. It's not easy to get out of Cuba anymore. And so Oriana, that I mentioned earlier, the Oriana Damas, and this is Alejandra Quintero, who's our research coordinator, and without her, we would be able to do none of this, did this study. She looked at, because I should tell you that one of the things that we did was in our database, in our IBD database, from the very beginning of our IBD database, we collected data on, where was someone born, what age did they come to this country, when did they develop symptoms, when were they officially diagnosed. Okay, so we had all that data, and so it allowed us to look at how old were they when they came, the mean age of immigration, how old were they when they got diagnosed, and that wasn't sort of statistically significant. But what was striking was the amount of time from immigration to diagnosis. And so whereas before 1980, right before that Mariel boat lift, when people came in, say, the 50s and all that, like my parents did, it took on average like 30 years for these people to develop IBD once they got to the United States. And then in this intermediate time between 1980 and 1994, the people who were able to immigrate to the United States took 17 years. Now, the mean time is 8 years. Now, that's a very striking difference and not a very huge difference in time. So it's amazing. I'm sure that all of you can have ideas of what that is. Is that only McDonald's? Is that something in the water? Is that, is what is happening? Is it getting jump started by changes that are happening perhaps on the island of Cuba itself? This is another one of my favorite studies. It is not related to IBD necessarily. It's just related to the concept of immigration and the change in the microbiome. And so these are Thai immigrants, but not just Thai immigrants, like Hmong and another tribe of like a very specific tribe of within Thailand. That's kind of important, maybe from a genetic perspective. I'm assuming less genetic diversity in that group, so there's less noise, right? And so pre-immigration, they have look how lovely and colorful their microbiome is in their gut. Then they get to LA, and they haven't even left LAX, and they're getting fat. One of them is getting fat. No, obviously they're coming here. I think if they went to the state of Washington up here, they'd probably stay thin, because I'm sure you guys are, eating healthier and all that jazz. But once they're more time in the United States, their gut microbiome diversity. This was, by the way, I should say, within 6 months. I was joking about LAX, but within 6 months, their microbial diversity starts plummeting. And then by the second generation, as their eyes start turning blue, people, the diversity is completely changed, and they're mirroring really what European Americans look like. So we're capable of upending people's gut microbiome very quickly if they decide to take the risk of coming to the United States, which, I've jokingly said up until now, that was Trump was trying to help people by building the wall. Okay, so I already mentioned this whole meconium study to you. This idea that when do we start, right? When does IBD start? That these children, these babies, newborns, already have altered microbiota at birth. And so these same investigators have paired with the University of Massachusetts, which have UMass has developed this anti-inflammatory diet. They've tested in IBD, and this is a kind of a cute study that we've modeled, a study that we're doing recently on this MELODY trial, where women who are pregnant and have Crohn's can voluntarily either just do whatever they're doing and send their stool, or will be taught about eating an anti-inflammatory diet and during pregnancy, I should say, during pregnancy, to see if that will change the microbiome of the newborn. So I think it's a really interesting study, and imagine the power of that you could start an intervention that is safe and healthy. Like, this, I don't know of anything that this would be could only be helpful for your heart, for everything, for cancer, for everything. So how could diet impact IBD, right? There's the effect of diet through the effect that it might have on the microbiome and the metabolome. Diet may directly affect inflammation. I'll show you one example of that. And then if you guys are eating something out of a bag, like, you opened a bag, or something that, I don't know, was frozen that you bought at whatever stores are out there. I don't know, Winn-Dixie, I don't know what's out there. There's a high likelihood it has preservatives, emulsifiers, microparticles that have also been accused of having a role in IBD. These are some of the things that have been associated epidemiologically with an increased risk of IBD: eating a high-fat diet, eating a high animal fat diet in particular. Fruits and vegetables protect against Crohn's and ulcerative colitis. And if you're a mouse, emulsifiers can aggravate the development of IBD. That's a little harder to prove in humans. We have emulsifiers in almost everything, and that has not been well quantified, even when you use things like apps to figure out what people are eating. This is an example to show you that in mouse models where you can have germ-free mice versus kind of standard mice that have like normal gut bacteria, there are foods that you can give a mouse, even in the germ-free state, that will exacerbate inflammation. So there's evidence that diet, some of the components in diet may actually have a directly detrimental effect, even in the absence of the intermediary step of the microbiome. We did a study that we recently published in CGH, I think October or November was when it officially came out, where we took ulcerative colitis patients that were in remission. We each person served as his or her own control, and they went through 4-week cycles of either a low-fat diet that was high in fiber or a high-fat diet that had intermediary levels of fiber. And we catered these diets because, for example, in the low-fat diet arm, we wanted only 10% of the calories from fat. I don't know what you guys are eating, in Seattle, but probably by breakfast you were done with the 10% calories from fat, because there's like a lot of fat in almost everything we eat, right? So we couldn't use the terminology high fat diet because our IRB thought that was unethical. So we called it SAD, for standard American diet. And then we had to add the ISAD, like we said, for idealized, because it turned out that we inadvertently gave them more fiber than they were actually eating at baseline. In other words, we didn't intend for it to be healthier, but it turned out that we inadvertently gave them a healthier diet. So here's an example of this. First of all, look at this fat intake at baseline in these patients that are in remission with their IBD, with their ulcerative colitis. I don't know, 30 plus % of their calories are from fat. More or less kept it that way for this SAD diet, and so that's okay. The low-fat diet by design, patients ate a lot less fat. Fiber intake at baseline is like nothing. Look at this, is servings of fruits and vegetables. Many people had zero intake of fruits and vegetables at baseline with ulcerative colitis in remission. So that's why when I say inadvertently, who knew that these people were eating no fruits and vegetables, almost no fiber. And so even in the SAD diet that we gave them a healthier diet. Yeah, I'll skip that one and tell you that, when we looked at bacterial diversity, when we went from the biggest change was when we went to a low-fat diet from their baseline diet. We found the biggest differences in the microbiota. We found some notable differences in some species of bacteria that are meant to be beneficial when we gave people a low-fat diet. I'll tell you that I think we had many fewer changes than what one might expect, but remember, it was a 4-week study. There's separation in metabolites. Again, this is only in a 4-week time frame, and I'll skip that for the sake of time. This is just a kind of a really cool way of representing the data that our bioinformatics people did, and this is to show anything that is sort of backed away from this outer circle is the activator, and on the other end is the recipient. So this is to really represent that diet has the biggest effect on the microbiome. Then it also has a big effect on metabolites, and then the microbiome has the biggest effect on metabolites. That would be a summary of that. One of the things that we found when we did this diet intervention study is that after we were done catering people's diet for 8 weeks, some people went crazy eating whatever they wanted to eat. They were like as if we had put them in a jail cell. We didn't put them in a jail cell. We just catered their diets and begged them to stay on it. So that led us to propose to the Helmsley Foundation a study that this holistic Crohn's diet study, which I'm very excited, and our team of people that I work with are so amazing and awesome. This is for Crohn's patients. The patients, in order to be in the study, have to have a significant other with whom they have meals over the age of 18. You'll see why. However, the patient gets to pick which arm of the study they will be in. They will either be they'll either just get diet counseling one time, or they will have their diet catered for 8 weeks, a low-fat diet, kind of reminiscent, but not quite as stringent as the one I just showed you for the ulcerative colitis patients. Or in the third group, the patient and a significant other, could be their mom, could be their boyfriend, girlfriend, whatever, someone that they live with. Both of those will get the diet catered, but in addition, they're going to get a psychological intervention, a dyadic psychosocial support program, where we have dietitians teaching them, like giving them knowledge about what they're eating and helping them to make a plan for how they will take what they've learned and now make the transition from having their diet catered to actually going to the supermarket and reproducing that in whatever it is that they enjoy eating. And we thought that was critically important, because if diet is going to be part of the armamentarium to either prevent or treat the disease, then we have to not only figure out what they should be eating, but how we're going to get them to eat that long term. I think you'll be so happy to know, this is my penultimate slide to say, in my conception of what we need to either cure or at the very least really have impactful treatments for IBD, it's going to require a multifaceted approach that takes into account what we know about the pathogenesis with respect to both immune response, epithelium, microbiome, and so, et cetera. And these are some of the great people that I get a chance to work with all the time. And this is us relatively recently. Julia Frisch, who just graduated, got her PhD in my lab, did a lot of the work on that diet intervention study. Okay, I'll shut up, Johnny. I'm going to stop sharing my screen.
No, that's great. You're more than welcome to share. We just have some questions coming in, and
Yeah.
maybe if there's any slides you want to go back to.
Okay, let me see my chat.
But first off, thank you so much. That was excellent, and you covered a lot, and I definitely learned a lot. So I think one of the earliest questions that came up, I think, was in respect to the fact that that study that you mentioned, where the time to diagnosis decreased in subsequent generations for immigration, and the question was there a possibility that the story was about our diagnostic capacity? Were we diagnosing this sooner, or do we feel like that this is a true sort of decrease in the time of onset?
Yeah, yes. I mean, we've had to kind of think through that question. The first is that we have been collecting data not only on the time of diagnosis, but when did the patient begin to have symptoms, and whether you looked at official diagnosis or time that they developed symptoms, they both tracked in the same direction. Mm-hmm. Moreover, ulcerative colitis is a bleeding disease. Like, you don't really develop ulcerative colitis and don't know there's something wrong with you. I accept that for Crohn's disease, that could be an issue. And we also stratified by those people that, we here at the university, we have a safety net hospital. We also have a kind of a private, a private clinic where patients have insurance. And whether we looked at it in the patients that were, if you will, uninsured, or those that have insurance, there was also not a difference, suggesting that it wasn't access to care and all that stuff. But, we continue to make diagnoses of IBD in people that have had vague symptoms, for a very long time. So mm-hmm. I think that's still possible.
Possibly part of it. Dr. Young had a question, which you should feel free to say yourself if you'd like to ask it. Yeah.
Maria, thank you. And thank you for this real tour de force on microbiome, IBD, immigration, and diversity. Thank you so much. Just a quick question, anecdotally, we're seeing people at home, they're snacking all day long, they stop brushing their teeth. There's a lot more tooth decay. I'm sure their microbiome will change. Any ideas what this whole COVID, is that going to change? Are you going to expect more IBD?
Wow. I don't know how much more can we have. It just seems it already seemed like an epidemic. I mean, from where I sit, it's like such an epidemic in immigrants. In a lot of these are like born in this country, kids that parents that are immigrants. I think that all this like hand sanitizer, washing your hands, wearing a mask, no spitting on each other, it has to be changing our microbiome. I even wonder if some of the antibiotic use that we plow on these patients that come in with COVID-19 will impact their immune response. There are some data, this is I guess tangential to your question, Barbara, but there is data in studies of the flu, for example, that antibiotic use may dampen antibody response to the flu vaccine. That's probably more true for those types of protein-mediated protein-based vaccines as opposed to, these mRNA kind of more complete immune response type vaccines. But I wouldn't doubt it that we'll see an increase. There are a couple of centers, and we will participate just as a site to follow these patients prospectively, at least our IBD patients prospectively, to see what their outcomes will be downstream of COVID, downstream of vaccination, what effect the drugs that they're on are having to immune responses to that. But your point is a great one. How might that change the epidemiology of it?
Thank you.
On a similar note, we talked a lot about bacteria, but there's plenty of viruses present as well. Yeah. And so I don't know if that has been looked at and doesn't seem to play as prominent a role, or if that's something that we're still looking at.
I wouldn't say, I think it hasn't been looked at as much, Johnny, like what you know. In a study that kind of got a bit of a flash, but you know how when like there's a flash, but then there are no subsequent studies from anyone, you sort of, don't know what to do with that. I mentioned briefly that this was a study done in a household, so that they could compare the index case to their other people in the household. People with IBD have less diversity. They had more bacteriophages, intestinal, bacteriophages that would theoretically be directed at gut bacteria. And why that's kind of cute is who's whoming who? Is it maybe these bacteriophages and viruses generally kind of depleting the microbiome that's having an effect? I think if you talk to Marty Blaser, Barbara will know who that is, but maybe someone else says someone's an old ID person might know who he is. But Martin Blaser, who is an ID person, but did very seminal work in Helicobacter pylori, and then sort of turned around and has contributed enormous amount to to understanding the human microbiome. And sort of discovering that we, through the use of antibiotics, are having radical impacts on our microbiome that we, just like this meconium study, now we see that, right, Johnny? Mm-hmm. We can't pass on certain microbiota to our newborns because the mom doesn't have it. And all that took is adding chlorine to the water and maybe filtering stuff and taking antibiotics, right? It'll have, and you don't get them back. And he and his wife are going to remote parts of the world. I hope they survive, because they're like going to the Amazon or to all these crazy places to collect stool and preserve it for to see what really the human bacterial diversity is, because they're not going to Seattle to figure that out, or coming to Miami. They want to look at it really in places that haven't been sterilized yet.
Well, I want to acknowledge that it's 1 p.m. And so thank you again, and thank the audience for being here and participating. There's a few more questions in the chat. If you've got 5 more minutes, I'd love to hear your thoughts.
It's 4:00 my time.
Well, thank you again. 4 o'clock on a Friday.
I'm delighted to be here. Remember, I would otherwise be rushing to the airport and trying to make a flight, right? So a couple more minutes with my friends here. Let me see. So where you found different bacteria in mucosal macrophages, were those associated with different cytokine responses? That's super important question. We found a couple of interesting things. The first is that the microbiota was similar in the human being. In other words, like if you biopsy that same person's ileum or colon, that microbiota was more similar in that individual, like ileum and colon, than it was across any individual. But wherever there was inflammation, wherever the biopsy was taken from an area of inflammation, those macrophages were expressing inflammatory cytokines and a lot of inflammatory markers. And our hope is, apropos to that very astute question, that now we may have, now the field may have enough of like a bioinformatic approach that if we sequence deep enough into like total RNA, we will be able to know what bacteria are there and simultaneously what the host immune response is. And I think that will be super cool. Did you see strongyloides as you see mimics in immigrants? This is my old school ID person. We have a lot of strongyloides here in our Latin American immigrants. We typically do screen for it, we do and have found people with strongyloides. I don't think it explains like a big like chunk of people that have IBD. Even when people have had other pathogens, when we treat them, they're still like, there's a lot of IBD once people come to this country. You also asked Wes about mycobacteria. I think everything is plausible. I really do think that it's plausible that mycobacteria, which are, you're an ID person, fussy, difficult to grow, hard to do functional studies. I don't think it's beyond reason to think that there's a subset of Crohn's. If you look at those pictures I showed you, it's like their tissues being eaten by something, right? So maybe in some people it is mycobacteria. It begs the question that how best to eradicate it, because actually just using antibiotics is maybe not good enough for those people that actually have this. The other thing about Koch's postulates that are difficult to fulfill in IBD is that it's equally conceivable that you could get rid of that mycobacteria and still like have enough sort of molecular mimicry that you won't get rid of the ongoing inflammation. So all those things I think are true and make it hard for the mafia. Like all these are factions. These are all factions, right? The mycobacteria faction that make it hard for them to prove or disprove it. And they're a very passionate group, which also kind of has an issue with reliability, like when people are a little overzealous.
I have a question, which is, you mentioned, are these two, ulcerative colitis and Crohn's disease, the different presentations along a spectrum of a single sort of homogeneous disease process, or are they heterogeneous, and should they be in two separate buckets? And sort of as the talk goes on, it seemed like you're bundling them together, but I'm just curious, explicitly, based on your experience, do you think that this will pan out, that there's really sort of different things going on and we've artificially bundled them and that precision medicine approach is going to arrive there? Or do you feel like what you're seeing is such an overlap in these gene loci that it's really sort of nuanced to how they got to where they are, but they all sort of having the same thing happen?
Yeah, that's a very provocative question. I think that the gut is the biggest interface that we have to the outside world.
Sure.
So it's not surprising that, just like in those mouse models where you just disrupt one thing and they get IBD, I think it highlights the fact that we need that whole process to be really dead on in order to control this kind of treacherous thing that we've done, which is say, okay, we're going to let a lot of bacteria that are typically pathogens, if you inhaled them, if you put them in your bladder, if you injected it into skin, you would have, you'd be septic, right?
Yeah.
But not when they're in the gut, right? So there's a very elaborate system to keep that in place, and tall fences make good neighbors. And so that long list of polymorphisms all contribute a little bit to this fence and kind of eroding this fence. And so you start with that as kind of a platform. But then when it comes to the actual expression of the disease, yeah, we've been studying genes, but these are cells that are doing this, right? Cells making things. And there's a big leap between what is the genetic polymorphism and how that is all interpreted, like all that information at the cellular level and at the level of sort of modules of cells kind of cooperating together to cause a problem. And I think that's where you either are going to get something that looks like ulcerative colitis, or you're going to get something that looks like Crohn's disease. And because it has to be something like that, Johnny. Epidemiologically, in an emerging population, ulcerative colitis starts rising, and then it's Crohn's disease. So that flip, you're not changing the gene. So what is that flip occurring from?
Mm-hmm.
Anyway, so those are all good questions for
a little bit of both, it sounds like. Yeah. Well, wow. Thank you so much. I would love to nerd out more on the individuals, like the PCA. It looked like the shift in the profile there between the lamina propria was more predominant if you were to draw those circles for Crohn's disease, which is a little different than what I would expect, since Crohn's disease seemed to have more genetic association. So it's like almost like at least what's going on in there may be the expression of those genetic differences instead of necessarily the driver. Like, I think that's so interesting. So I'm excited to see how your mentees continue to progress this story. Thank you so much for being with us. And thank you to everyone in attendance, and have a great Friday.
All right. Well, have a wonderful 21, guys. Okay. Thanks for having me as your first speaker this year.
Thank you for
Same to
bringing in the new
you.
year.
Thank you, Maria.
Bye. Bye.
Bye.